Ever had that moment? You know, you stub your toe, maybe a paper cut starts to sting a bit. It feels
Mm,
pretty minor, right? But underneath, there's this whole microscopic drama unfolding.
that's exactly it. And that immediate reaction, the heat, the discomfort, that's your body's fundamental response.
Yeah.
Inflammation.
Inflammation.
Yeah.
Yeah, it's a word you hear constantly, often in a negative way, like anti-inflammatory everything.
Right.
But today, we're really going to dive deep. We've got these scientific sources, and we want to unpack what inflammation really is.
Uh-huh. Going beyond just, the basics of redness and swelling.
Exactly. We're talking molecules, cells working together,
Yeah.
the whole picture.
Yes. Our mission really is to get into the key mechanisms, understand its importance, not just as a symptom, but as this core protective and healing process. We've looked at some quite detailed scientific texts exploring all these interactions.
Cells, proteins, how they orchestrate everything we actually see and feel.
Okay.
And for you listening, if you want to get a solid handle on something complex like this, but, without feeling totally overwhelmed,
Yeah.
this is kind of your fast track. We're aiming to pull out the essentials, connect the dots clearly, maybe have a few aha moments along the way.
Hopefully.
Okay, so let's start right at the beginning. In the grand scheme of things, what is inflammation?
Well, think of it as your body's first responders, its initial, very organized defense system.
Okay.
It gets triggered by all sorts of physical damage, like that stubbed toe,
Right.
chemical irritants or invaders, bacteria, viruses.
And we recognize it by those classic signs.
Exactly. Heat, redness, swelling, and pain. Kaler, Ruber, tumor, dolor. They've been recognized for centuries, actually.
Kaler, Ripper, tumor, dolor. Okay. But what's actually causing that heat and redness and swelling? What's happening inside?
So those are the direct results of changes in your blood vessels
Mm.
and what we call fluid and cell leakage or exudation.
Yeah.
Yeah. In that affected area, the blood vessels widen vasodilation. That increases blood flow, causing the redness and the heat.
Makes sense.
And at the same time, those vessels become more permeable, sort of leakier. This lets fluid and immune cells move out into the tissues around them.
Hence the swelling, the tumor.
Exactly.
Yeah.
And then various chemical signals get released, stimulating nerve endings. And that's where the pain, the dolor, comes from.
So it's like the body flooding the zone, sending in the troops and supplies right where they're needed.
A good analogy, yes.
But we hear so much about inflammation being bad. Is it always a good thing?
That's a really critical point.
Mhm.
Inflammation is it has a dual nature. It's absolutely vital. We need it to fight infections, to repair damage.
Right. Healing wouldn't happen otherwise.
Precisely. But if that response is too strong, or if it just keeps going and going, it can be harmful. It can contribute to a whole range of chronic diseases.
So it's a balance, a really fine balance.
A very fine balance, yes.
Yeah. Okay, that makes sense. Now, our sources dig into some specific molecular players. They start with arachidonic acid and prostaglandins. Sounds very sciencey.
They do sound complex, but arachidonic acid is basically a fatty acid. It's a key starting material for the whole inflammatory process.
Okay, a building block.
Sort of. When your cells are damaged or activated, certain enzymes go to work on this arachidonic acid, converting it into various signaling molecules. And a really important group of these are the prostaglandins, or PGs.
PGs. Got it. And what do they do when inflammation kicks off?
They do a lot.
Mm.
In inflammation, they contribute to that vasodilation we mentioned, the widening of blood vessels. They're also major players in causing pain and fever.
Okay. Pain and fever, common inflammation symptoms.
Right. But here's where it gets really interesting, kind of an aha moment maybe. Prostaglandins aren't just about inflammation.
Oh.
They have other jobs, seemingly totally unrelated jobs, elsewhere in the body, like in your stomach.
The stomach.
Well, they actually help stimulate the production of protective mucus and, at the same time, reduce stomach acid secretion.
Wow. Okay. So the same thing causing pain in my stub toe is protecting my stomach lining.
Exactly. Quite the contrast, isn't it?
It really is. What else? Any other surprising roles?
Yep. They're involved in the female reproductive system, too. Specifically, one type, PGF2 alpha, seems to play a role in breaking down the uterine lining during menstruation.
Huh.
And, maybe even more surprisingly, prostaglandins stimulate uterine muscle contractions during labor.
That's incredible. So this one pathway, starting with this fatty acid, leads to molecules that cause inflammation, protect the stomach, are involved in periods, and help with childbirth.
That's right. It really shows how interconnected biological systems are, how evolution kind of repurposes these fundamental tools for different jobs.
Amazing. Okay, so you have these molecules, but something needs to trigger them, right? How does the body even know there's a threat?
Great question. It needs danger signals. Our sources talk about PAMPs and DAMPs.
PAMPs and DAMPs, right. What are they?
Okay, so PAMPs are pathogen-associated molecular patterns. These are molecules typically found on microbes, viruses, bacteria, fungi, but not usually on our own cells.
Like their signature.
Exactly. Think of things like double-stranded RNA from viruses,
Mm.
or lipopolysaccharides, LPS, from bacteria, or beta-glucans from fungi. They're like microbial calling cards.
Got it. Foreign invaders. What about DAMPs then? Damage associated.
Damage-associated molecular patterns, yes. These are molecules released from our own cells when they're damaged or stressed out.
Okay, so internal alarm signals.
Right. Things like HMGB1, ATP, uric acid, even heat shock proteins. So whether it's an invader or just damage to our own tissues, the body has ways to sense, uh-oh, something's wrong here.
It's like a built-in surveillance system. And how are these PAMPs and DAMPs actually detected? What's doing the sensing?
That's where receptors on our immune cells come in. A major group are the toll-like receptors, or TLRs.
TLRs.
Different TLRs recognize different PAMPs. For instance, TLR4 is famous for recognizing bacterial LPS. TLR3 detects that viral double-stranded RNA.
Specific detectors for specific threats?
Exactly. And there are others too, like RAGE receptor for advanced glycation end products, which recognizes various DAMPs.
So these receptors are like the security guards patrolling and looking for trouble signs, and when they spot a PAMP or a DAMP, boom.
Pretty much. That recognition flips a switch inside the immune cell, triggering signaling pathways that kickstart the whole inflammatory response. It's the go signal. Activate defenses, call for backup.
And the backup is the immune cells, the leukocytes.
Exactly. The leukocytes, or white blood cells.
Mm-hmm.
They are absolutely central. They need to get out of the bloodstream and into the tissues, right to the site of the problem.
Right.
And that process is called leukocyte extravasation. It's not random. It's a very specific multi-step journey.
Okay, break that down for us. How do they physically get out of the blood vessels?
It starts with rolling. The endothelial cells line the blood vessels, start expressing these adhesion molecules called selectins. There's P-selectin, E-selectin, L-selectins.
Selectins.
Okay. Leukocytes have carbohydrates on their surface that bind, but only weakly, to these selectins. So the leukocytes slow down and sort of tumble or roll along the vessel wall.
Like hitting the brakes gently?
Kind of. And interestingly, things like histamine and thrombin can quickly make endothelial cells display P-selectin, while cytokines like TNF and IL-1, which we'll get to, trigger E-selectin expression later. It shows how early signals control this recruitment.
Okay, so they're rolling along. How do they stop and actually stick?
That's the next step, firm adhesion. Leukocytes have other adhesion molecules called integrins. These bind much more strongly to complementary molecules on the endothelial cells, like ICAM1 and VSICM1.
Stronger binding, like Velcro.
Exactly like Velcro. And signaling molecules called chemokines, released at the inflammation site, are crucial here. They basically activate the integrins, making them much stickier.
Okay, stuck fast. Now, how do they get through the vessel wall?
That's transmigration, or diapedesis. Once they're firmly attached, leukocytes literally squeeze themselves between adjacent endothelial cells.
Yes.
Yeah, it involves other molecules like PCAM1, acting almost like zipper guides. They push through the junctions into the tissue.
An incredibly precise delivery system for the immune cells?
Absolutely.
Yeah.
Target acquired, troops deploying. And once they arrive, they don't just sit there. They start releasing a whole barrage of their own chemical signals, inflammatory mediators.
Amplifying the response.
Exactly. Amplifying it, controlling it, recruiting more help. It's a complex communication network.
Okay, this sounds like where it gets really complicated. The sources list tons of these mediators. How can we organize them?
Well, we can think about them in two main groups.
Mm-hmm.
Those released directly from cells at the site,
Okay.
and those derived from proteins that are already circulating in the blood plasma, just waiting to be activated.
Got it. Let's start with the cell-derived ones.
Okay, histamine is a big one. Comes from mast cells, basophils, platelets. We know it from allergies, right?
Yeah.
It causes vasodilation, widening vessels, and increases vascular permeability, making them leaky, contributes to swelling, and it activates those endothelial cells lining the vessels.
Okay, histamine. What else?
Serotonin, mainly from platelets, does similar things to blood vessels. Then we have those prostaglandins, again, PGs, causing vasodilation, pain, fever.
Right, we covered those.
Then there are leukotrienes, also derived from arachidonic acid, but via a different enzyme pathway.
Okay.
They also increase permeability, can constrict airways important in asthma, and they're chemotactic,
Chemotactic,
meaning
meaning they attract other immune cells to the area, like leaving a chemical trail for others to follow.
okay. So lots of signals, some overlap, some distinct functions. What else from the cells?
There's platelet activating factor, PAF, very potent, does a lot. Clumps platelets, constricts or dilates vessels depending on context, constricts airways, increases permeability, helps leukocytes stick, attracts leukocytes, triggers degranulation. Wow, a real multitasker.
Definitely. Then reactive oxygen species, ROS, produced by neutrophils and macrophages, great for killing microbes, but they can also damage your own tissues if there's too much collateral damage.
Right.
Similar story for nitric oxide, NO, produced by endothelial cells, macrophages, causes vasodilation, helps kill microbes. Again, different enzyme versions exist called NOS, isoforms, ENO, NNOS, NO, used in different situations.
It's incredible the level of chemical signaling involved.
It really is. And we absolutely have to mention the major cytokines,
Yeah.
like tumor necrosis factor alpha, TNF, and interleukin-1, IL-1,
TNF and IL-1, heard of those?
key orchestrators made by macrophages, T cells, others. Locally, they activate endothelial cells, ramp up leukocyte recruitment.
And systemically, affecting the whole body.
Yes, they cause fever, tell the liver to make acute phase proteins, and can even cause that lack of appetite, the anorexia you feel when you're sick.
Ah, so that's why you don't feel like eating.
Partly, yes. And finally, in this cell-derived group, we have the chemokines, a huge family of small proteins, their main job. Chemotax is attracting specific types of leukocytes.
This specific come here signals.
Exactly. For example, CXCL8, also called IL-8, is a powerful magnet for neutrophils, brings them right in.
Okay, that's a lot from the cells. What about the ones from plasma proteins in the blood?
Right, these systems are circulating, ready to be activated. Three main ones, complement, coagulation, and Keening systems.
Complement system, what does that do?
It's a cascade of proteins. Can be activated in three ways: classical, alternative, lectin pathways. Key functions include opsonization,
Opsonite.
tagging microbes, like putting eat me signs on them for phagocytes.
Got it.
It also produces anaphylatoxins, C3A and C5A. These cause histamine release from mast cells and are also chemotactic, attracting more cells.
More attraction signals.
Yes. And finally, it forms the membrane attack complex, the MAC. This literally punches holes in microbial membranes, killing them directly. A multi-pronged attack system.
Powerful stuff. And the coagulation system, isn't that just for blood clots?
Primarily, yes, it forms fibrin clots, but activating it also spins off other inflammatory molecules. Fibrinopeptides, for example, increase vascular permeability and are chemotactic. And thrombin, the enzyme that makes fibrin, also activates endothelial cells.
So clotting and inflammation are linked.
Very much so. And the third one, the kinin system.
What about that?
This system generates bradykinin. It's cleaved from a precursor protein, HMWK, by an enzyme called kallikrein.
Bradykinin, okay.
And bradykinin is potent, increases vascular permeability, causes vasodilation, and is a major contributor to pain and inflammation.
Ah, a key pain molecule.
Definitely one of the main culprits for why inflammation hurts so much.
Okay, so we have the triggers, the signals flying everywhere. Let's focus on the cellular soldiers now. The leukocytes doing the work.
Right, the specific cell types are crucial. Neutrophils are usually the first ones in, the pioneers of acute inflammation.
Acute being short term.
Yes, short term. Neutrophils are phagocytes that eat microbes and debris, and they release nasty stuff stored in their granules like enzymes and those ROS molecules to kill invaders. Think of them as the shock troops, the initial cleanup crew.
First on the scene, aggressive. Who follows them?
Macrophages. They play a much broader role in both acute and, importantly, chronic inflammation.
Chronic is the long-lasting kind.
Exactly. Macrophages are also phagocytic. They produce cytokines like TNF and IL-1, but they also act as antigen presenters.
Meaning?
They show pieces of the invader's antigens to T lymphocytes, bridging the gap between the initial innate response and the more specific adaptive immune response.
Connecting the dots for the immune system.
Precisely. And macrophages aren't monolithic. They can be activated in different ways. M1, or classically activated, are pro-inflammatory, good at killing microbes. M2, alternatively activated, are more involved in cleaning up, tissue repair, dampening inflammation. Different jobs for different phases.
So they adapt their role. More versatile than neutrophils. What about eosinophils and mast cells, often linked to allergies, right?
That's right. Eosinophils are key players against parasitic worms and also central in allergic reactions. Their granules contain things like major basic protein, toxic to parasites, but also damaging to our own tissues in allergies, like in asthma.
And mast cells.
Also critical in allergies, those immediate hypersensitivity reactions. They sit packed with granules full of histamine and other mediators, ready to release them instantly when triggered. That's why allergic reactions can happen so fast. Itching, swelling, wheezing.
Instant release. Okay. And lastly, lymphocytes, T cells and B cells. Where do they fit?
They are the stars of the adaptive immune system. Less prominent right at the start of acute inflammation, but absolutely critical for chronic inflammation and for providing long-term specific immunity.
How do they contribute to inflammation itself?
T cells produce cytokines that can either fuel or regulate inflammation. B cells develop into plasma cells that produce antibodies, specifically targeting pathogens. They bring memory and specificity to the fight, especially in longer battles.
Okay, so we've got the triggers, the signals, the cells. What happens at the end of an acute inflammatory episode? What are the outcomes?
Ideally, resolution. The injurious agent is gone, the mediators fade, the cells are cleared out, the tissue goes back to normal. Happy ending.
Best case. What else?
If there's been substantial tissue destruction or the body can't fully repair the original structure, you get fibrosis or scarring. Collagen gets laid down, patching the area, but function might be impaired.
A scar instead of full repair.
Right. And the third outcome is progression to chronic inflammation. This happens if the initial cause persists or the immune system just doesn't switch off properly. The inflammation becomes ongoing.
Stuck in on mode. Which leads us to chronic inflammation. How is it distinct from acute?
The main thing is duration weeks, months, years. And the cellular picture changes. Instead of neutrophils dominating early on, chronic inflammation features more lymphocytes and macrophages.
The adaptive immune players and the versatile macrophages?
Exactly. And their continued presence, releasing mediators, leads to ongoing tissue injury, attempts at repair, often alongside fibrosis. It's a simmering, damaging state.
What typically causes this chronic state?
Several things. Persistent infections the body can't eliminate, like certain viruses or mycobacteria. Autoimmune diseases where the immune system mistakenly attacks self-tissues, think rheumatoid arthritis. Or prolonged exposure to toxins or irritants you can't easily get rid of.
Makes sense. The sources also mention a specific pattern, granulomatous inflammation.
Yes, that's a distinctive type of chronic inflammation. It's characterized by collections of activated macrophages, often looking quite different. They're called epithelioid cells because they resemble epithelial cells.
Okay.
These collections are often surrounded by lymphocytes. It's basically the body's attempt to wall off something it finds really hard to eliminate.
Like building a barrier around the problem.
Exactly. You see it in tuberculosis, where the body tries to contain the bacteria,
Yeah.
or in response to foreign materials like splinters or sutures that can't be broken down. You might also see giant cells within these granulomas langens, giant cells, or foreign body giant cells, formed by macrophages fusing together.
Fused macrophages trying to engulf something large or persistent.
That's the idea, a specialized containment strategy.
Wow. Okay, this has been quite the journey through inflammation. It's way more intricate, more interconnected than just redness and swelling.
Absolutely. It's a truly remarkable biological process. This constant dynamic interplay between molecules and cells, all aimed at protection and healing.
But with that potential downside, that delicate balance we talked about,
Precisely. When it's well regulated, it's essential. When it goes awry, it's involved in so many diseases.
so maybe the next time you feel that little twinge of inflammation, you can appreciate the sheer complexity going on under the skin. Think about that balance and how crucial it is for our health when it works right, and how problematic when it doesn't.
It really underlies so much of health and disease. And honestly, what we've covered today is still just scratching the surface.
There's more.
Oh, absolutely.
Yeah.
The signaling pathways inside the cells, the specific mechanisms that actually resolve inflammation and shut it down. There's so many more layers.
Well, hopefully this deep dive has sparked some curiosity for you listening. If any part of this caught your interest, we definitely encourage you to explore it further. It's a huge area of research with massive implications.